Chapter 20. Biotechnology and Genetic Modification. In this chapter you will find out how we can use microorganisms and enzymes to make useful products. Learn about some examples of genetic modification. Explain how genetic modification is done. Discuss the advantages and disadvantages of genetic modification of crop plants. Getting started. With a partner, write down at least 2 facts about DNA. Take turns with other pairs in your class to write one of your facts on the board. Keep going until all the different facts have been added. Golden rice. Humans have been altering the genetic makeup of organisms for thousands of years, long before we knew anything about chromosomes, genes or DNA. People began growing wheat and other cereal crops more than 11,000 years ago. Archaeological evidence shows how the crops changed over time as people used the natural variation in the crop plants and selected the best seeds to sow. They also used selective breeding to improve the animals they kept for meat, milk, skins and to pull their carts and plows. In the 20th century, we learnt more about genetics and began to use our knowledge of genes to plan how to breed plants or animals to get the particular features that we wanted. In 1953, Watson and Crick announced that genes are made of DNA. Since then, scientists have learnt how to move lengths of DNA from one cell to another. Now we are even able to alter the DNA in a cell. Not everyone is comfortable with this. For example, in the 1990s, a project was begun to put DNA from other organisms into rice plants so that the plants make an orange substance called carotene, which the human body can convert to vitamin A. Vitamin A deficiency causes an estimated 4,500 deaths a day, mainly in children, and also causes blindness. The scientists successfully produced genetically modified rice, called golden rice, which contains much more carotene than normal rice. But so far, no one has used it, mostly because many governments and individuals have been worried about possible dangers of GM crops. Eventually, by 2019, Australia, Canada, New Zealand and the USA had registered this rice as safe, and many developing countries will probably follow their lead. The rice should be no more expensive for farmers to grow than ordinary rice, and it could save the sight and lives of huge numbers of people. Discussion questions. 1. Why do you think that people are suspicious of genetically modified organisms, such as golden rice, but have no problem in accepting crops or animals that have been produced by selective breeding? 2. Do you think that governments concerned about the possible dangers of GM crops should ban them, or are the potential life-saving benefits more important? Figure 20.1, a researcher working with golden rice plants in a glasshouse. 20.1 Biotechnology. Biotechnology involves using living organisms to carry out processes that make substances that we want. Usually, the term is used only when microorganisms are involved, or when plants or animals are used to produce something other than food. We have been using microorganisms to make various products for us for thousands of years. Yeast has been used to make bread and alcohol. Bacteria have been used to make yogurt and cheese. Of course, people did not know that these microorganisms were involved in the processes they used because they are too small to see with the naked eye. Today, we still use microorganisms to make these foods, but we now also use them to make many other substances, such as enzymes. And, in the 1970s, a new branch of biotechnology began when scientists first found out how to take a gene from one organism and put it into a different one. This is called genetic modification, and it has opened up entirely new possibilities for using microorganisms and other organisms. Using microorganisms. Biotechnology and genetic modification often make use of microorganisms, such as bacteria. There are several reasons for this. Bacteria are very small and are easy to grow in a laboratory. They do not take up a lot of space. Bacteria reproduce very quickly. They are able to make a huge range of different chemical substances. Bacteria can make complex molecules that are useful to humans. No one minds what is done to bacteria and fungi. There are few ethical issues like those that might arise if we used animals. As well as their main DNA, their chromosome, bacteria also have little loops of DNA called plasmids. These are quite easy to transfer from one cell to another. We can use plasmids for moving genes from one organism's cells to another. Making use of anaerobic respiration in yeast. Yeast is a single-celled fungus. Figure 20.2 shows yeast cells seen using an electron microscope. Figure 20.2, yeast cells seen with a scanning electron microscope. You can see little buds growing from some of the cells. This is the way that yeast reproduces. Yeast is able to respire anaerobically. When it does so, it produces ethanol and carbon dioxide. Glucose ethanol plus carbon dioxide making ethanol for biofuels. In Chapter 19, you saw that we need to reduce our use of fossil fuels. One alternative is to use plants to provide sugars, which yeast can then break down to form ethanol. The ethanol, sometimes called bioethanol, can then be used as a fuel. Maize and sugarcane are two of the crops that are used in this process. Maize is first treated with amylase enzymes, which break down stored starch to glucose. Sugarcane does not need to be treated with enzymes because it stores most of its carbohydrate as sucrose rather than starch. Yeast is then added and allowed to use the glucose in anaerobic respiration. The ethanol that is produced can then be extracted from the mixture by distillation. Although alcohol burns well, it does not contain as much energy per liter as fossil fuels. It is therefore normally mixed with gasoline to make biofuel that is used in vehicle engines. A possible advantage of using a biofuel like this is that the ethanol used is a sustainable resource. We can keep growing more maize to make more fuel. It also helps to reduce the amount of carbon dioxide that we add to the atmosphere. Although carbon dioxide is produced when the biofuel is burnt, the plants that are grown to make the fuel will have recently taken in carbon dioxide from the air during photosynthesis, using it to make the sugar and starch. This contrasts with burning fossil fuels, because then we are releasing carbon dioxide into the air that has been stored in the earth for millions of years. However, there are arguments against growing crops to make biofuels. These crops take up land that could otherwise be used to grow food for people. Using large quantities of maize and other crops to make biofuels puts up their price, making it more expensive for people to buy food. The land on which the crops are grown may originally have contained a biodiverse natural ecosystem, which was cleared to increase the area for growing the monoculture crop. Figure 20.3, sugarcane being unloaded at a factory in Brazil. The sugarcane is used to make sugar, and the leftover waste material is used to make ethanol for biofuel. Activity 20.1. Thinking about biofuels. Some people think that using biofuels instead of fossil fuels can help to reduce climate change. Others think that biofuels are not a good answer to this problem. Take a few minutes to think about your own view on this issue. Then turn to a partner and share your ideas. When every pair is ready, take turns to share your point of view and your reasons for it with the rest of the class. Reflection. How satisfied are you with your contribution to the discussion about biofuels? How clearly do you think you explained your point of view? Did you express yourself clearly? How successfully did you back up your argument with facts and examples? In future, what can you do to improve the way that you contribute to a discussion such as this? Making bread. Bread is made from flour, which is made by grinding the grains of cereal crops. Most bread is made from wheat flour. Flour contains a lot of starch and also protein, especially a protein called gluten. To make bread, the flour is mixed with water and yeast to make dough. Amylase enzymes break down some of the starch in the dough to make maltose and glucose, which the yeast can use in anaerobic respiration. It produces bubbles of carbon dioxide. These get trapped in the dough. Gluten makes the dough stretchy, so the carbon dioxide bubbles cause the dough to rise. Anaerobic respiration also makes alcohol, but this is all broken down when the bread is baked. Baking also kills the yeast. Figure 20.4, yeast has been respiring anaerobically in this pizza dough. The carbon dioxide produced by the yeast made the dough rise. Questions. 1. State 2 reasons why microorganisms, rather than animals, are often used in biotechnology. 2. Which product of anaerobic respiration by yeast is used to make biofuels? 3. Which product of anaerobic respiration by yeast is important in bread making? Making use of enzymes. Many different enzymes are used in industry. Most of them are obtained from microorganisms. Pectinase. Fruit juices are extracted using an enzyme called pectinase. Pectin is a substance which helps to stick plant cells together. A fruit such as an apple or orange contains a lot of pectin. Pectinase breaks down pectin. This makes it much easier to squeeze juice from the fruit. Pectinase also helps to make the juice clear rather than cloudy. The cloudiness in fruit juice is often caused by insoluble carbohydrates that float in the juice. Pectinase and other enzymes such as cellulase can be added to the cloudy juice. They break the insoluble substances to soluble ones, so the juice becomes clear. Figure 20.5, the apple juice in the left-hand bottle has been treated with pectinase to break down insoluble pectin that makes the juice in the right-hand bottle look cloudy. Biological washing powders. Washing powders contain detergents. These help insoluble fats and oils mix with water so they can be washed away. Biological washing powders contain enzymes as well as detergents. These enzymes help to break down other kinds of substances which can stain clothes. They are especially good at removing dirt which contains colored substances from animals or plants, like blood or egg stains. Some of the enzymes are proteases which catalyze the breakdown of protein molecules to amino acids. This helps with the removal of stains caused by proteins, such as blood stains. Blood contains the red protein hemoglobin. The proteases in biological washing powders break the hemoglobin molecules into smaller molecules which are not colored and which dissolve easily in water and can be washed away. Some of the enzymes are lipases which catalyze the breakdown of fats to fatty acids and glycerol. This is good for removing greasy stains. To prevent these enzymes from digesting proteins and fats in the skin of people handling them, the enzymes are packed into microscopic capsules. The capsules break open when the washing powder is mixed with water. Figure 20.6, scanning electron micrograph of granules of biological washing powder. The enzymes are packed inside the microscopic granules. Experimental skills 20.1. Investigating biological washing powders. You are going to work in a small group to plan and carry out an experiment to investigate whether or not biological washing powders are better at removing stains than other washing powders. You will practice writing a careful plan before you do your experiment. As you work, you will have the chance to make changes to your plan if you can see how it could be improved. You will need, for writing the plan, a pencil and paper. For your experiment, the apparatus and materials that you decide to use. Method Part 1, Writing the Plan. Here are some hypotheses that you could investigate. Choose one of them. Alternatively, you could write your own hypothesis. It is best if each group in the class investigates a different hypothesis. Biological washing powders remove egg stains better than non-biological washing powders. The optimum temperature for biological washing powders is lower than the optimum temperature for non-biological washing powders. Biological washing powders are better than non-biological washing powders at removing stains from cotton cloth. Then, as you write your plan, think about these points. What is your independent variable? How will you change it? How many different values will you use? What will be your highest and lowest value? Note, you may want to do some preliminary work to decide on suitable concentrations or masses of washing powder to use. What is your dependent variable? How and when will you measure it? What variables do you need to keep the same? How will you do this? What apparatus will you need? What sizes of glassware will you use? What measuring instruments do you need? How many? Are there any risks involved? What will you do to keep yourself and others safe? What do you predict your results will show? Why do you think this will happen? Now make your list of what equipment you will need to carry out your plan. Safety. Make notes on any safety issues you can think of. Remember, you are dealing with biological washing powder. Method Part 2, Doing your experiment. As you work, you may decide to make changes to your original plan. This is fine, it is a good thing to do. Record and display your results in the way that you think is best. Questions. 1. What problems did you have in carrying out your experiment? How did you solve them? 2. How well did your results match your prediction? 3. If you did the same experiment again, would you expect to get exactly the same results? Explain your answer. 4. What do you think are the main sources of error in your measurements and results? How could you reduce these so peer assessment? When everyone has completed their experiment, each group in the class should present a short description of their experiment and their results and conclusion to the rest of the class. As you listen to the first group's presentation, write down 2 things that you think they have done really well. Also write down 1 thing that you think they could improve and explain how they could improve it. The class can then give feedback to the group before the next group gives their presentation. Questions. 4. Explain why pectinase helps to obtain more juice from fruit. 5. What do biological washing powders contain, as well as detergents? 6. The first biological washing powders only worked in warm, rather than hot, water. Suggest why. 7. The enzymes that are used in modern biological washing powders are often obtained from bacteria which naturally live in hot water, in hot springs. Outline how natural selection may have resulted in the adaptation of these bacteria to live in hot springs. Lactase. Lactase is an enzyme that breaks down lactose, the sugar found in milk. All human babies produce lactase in their digestive systems. This is needed to help them to digest the lactose in the milk that is their only source of food for the first few months of their life. Some people, for example, those of European descent, continue to make lactase all of their lives. However, most people, for example, those of Asian descent, stop making lactase when they are adults. These people cannot digest lactose. They may feel ill if they eat or drink foods that contain lactose. Milk can be treated with lactase to break down the lactose so that people who don't make lactase themselves can drink milk or eat products made from the lactose-free milk. Figure 20.7, cow's milk that has been treated with the enzyme lactase can be safely drunk by people who do not make lactase in their digestive systems. Another reason for treating milk with lactase is to produce the sugars glucose and galactose. These can be used for making sweets. Growing microorganisms in a fermenter. When bacteria or yeast are used to produce enzymes or other substances for our use, they are grown in a container called a fermenter. This allows production of the useful products on a large scale. Figure 20.8 shows a typical fermenter. Figure 20.8, a fermenter for growing microorganisms. Sterilization. Before the fermenter is used, it is thoroughly washed and sterilized. This is usually done by passing steam through it. This kills all the microorganisms that are in the fermenter so that they will not contaminate the culture. Providing nutrients. Water and the nutrients that the microorganisms need for growth are added to the fermenter. The exact nutrients required depend on the microorganism that is being cultured and the product that we want. Glucose may be added, which the microorganisms can use in respiration to provide them with energy. Amino acids or nitrate ions may be added to provide a source of nitrogen so the microorganisms can make proteins. Providing air. If the microorganisms will respire aerobically, air is bubbled through the contents of the fermenter. This provides oxygen. The fermenter has an outlet through which waste gases can escape to make sure that the pressure inside the fermenter does not build up. Mixing the contents. In most fermenters, a motor turns paddles that keep the contents mixed. However, if the fermenter is being used to grow a fungus made up of long hyphae, there are usually no paddles because they would get tangled up with the hyphae. Air bubbles also help with mixing the contents. This makes sure that the microorganisms in every part of the fermenter are supplied with nutrients and oxygen. Adding the microorganisms. When all this has been done, the microorganisms are added. Measuring and maintaining temperature. A temperature probe measures the conditions inside the fermenter. Temperature must be kept at the correct level so that the enzymes in the microorganisms can work effectively to produce the required product. As the microorganisms respire, they release heat energy, which can cause the temperature to increase above the optimum for their enzymes. Cold water is then passed around the fermenter to lower the temperature. If, however, the temperature falls below the optimum, then hot water or even steam can be passed around the fermenter. Measuring and maintaining pH. The pH of the fermenter contents is measured using a probe. It is important to keep the pH at the optimum level for the microorganisms' enzymes. If the microorganisms are producing carbon dioxide, this will lower the pH. pH is adjusted by adding small quantities of acidic or alkaline liquids to the contents of the fermenter. Harvesting the product. When the microorganisms have had time to produce the required product, the contents of the fermenter can be harvested. This may be done by allowing some of the contents to run out through an outlet tube, or sometimes the whole fermenter is emptied at once. Purifying the product. The contents of the fermenter include the required product, but there will also be other substances, including the microorganisms themselves, which are not wanted. The required product is now separated from the unwanted substances so that it can be used or sold. Many different substances are now produced commercially using fermenters. These include insulin, made by genetically modified bacteria; the antibiotic penicillin, produced by the fungus Penicillium; a food called mycoprotein, produced by a fungus called Fusarium. Questions. 8. Explain why lactose-free milk is produced. 9. Explain why it is essential to sterilize a fermenter before it is used to make a product. 10. What circumstances might it not be necessary to bubble air into a fermenter? 11. One product that is made in fermenters is the antibiotic penicillin. This is made by a fungus called Penicillium. The fungus is made up of long threads called hyphae. A. Explain what an antibiotic is used for. B. Carbohydrates and amino acids are added to the fermenter. Explain why both of these substances are added. C. The fermenters in which penicillium is cultivated do not usually have paddles. Suggest why. 20.2 Genetic modification. We have seen that a gene is a length of DNA that codes for the production of a particular protein in a cell. Genetic modification involves changing the genetic material of a cell or of a whole organism. This can be done by removing genes, altering genes, or adding individual genes to cells. Genetic modification was first carried out in the 1970s. Since then, many different uses have been found for this process. Making human proteins. Human genes can be inserted into bacteria. The bacteria follow the instructions on the gene to make a protein that is normally made in human cells. For example, insulin is now produced by GM bacteria. The human insulin gene is inserted into the bacteria, and the bacteria are grown on a large scale. The insulin that they produce is purified. The insulin made by these bacteria can be used by people with type 1 diabetes who need to inject insulin regularly. Before GM insulin was available, insulin had to be extracted from the pancreases of dead animals, such as pigs. Producing it from GM bacteria enables much more insulin to be produced in a short period of time, and also ensures that the insulin is pure and of a consistent quality. Making herbicide-resistant crop plants. Herbicides are sprayed on crop plants to kill weeds. However, this is no use if the herbicide also harms the crop plants. Some crop plants have been genetically modified to be resistant to herbicides or insect pests. For example, soya plants have been genetically modified so that they are not harmed when a herbicide called glyphosate is sprayed onto them. This means that farmers can spray a field of these plants with the herbicide and only the weeds are killed. Making pest-resistant crop plants. Each year, huge quantities of crops are damaged by insect pests. This can greatly reduce yields. Farmers spray pesticides to kill the pests, but this is expensive, and the pesticides kill useful or harmless insects as well as the pests. Some crop plants have been genetically modified so that they are not damaged by insect pests. For example, cotton plants have been genetically modified so that they contain a substance called Bt, which is toxic to insects. Insect pests, such as the cotton boll weevil, are killed if they eat the cotton plants. This reduces the use of pesticides. Figure 20.9, cotton boll weevils are beetles whose larvae live inside the fruits of cotton plants. They are very destructive and can greatly reduce the harvest of cotton from the plants. Improving nutritional qualities of crop plants. Some crops have had genes inserted into them that cause them to make larger quantities of nutrients that humans need. At the start of this chapter, we saw that lack of vitamin A is a big problem for children in some parts of the world. This is especially so where the diet mainly consists of white rice. Severe vitamin A deficiency can cause blindness and is thought to kill more than 1 million people each year. Genes from maize and bacteria have been inserted into rice, and the rice uses these genes to produce a substance called carotene. The human body can turn carotene into vitamin A. Carotene is the substance that makes carrots look orange, so the rice grains are yellow. This GM rice is called golden rice. Figure 20.10, where white rice makes up most of the diet of children, using golden rice instead could greatly reduce the risk of vitamin A deficiency. Activity 20.2. Should food containing GM crops be labeled? Europe, the USA and some other countries, food that contains nutrients derived from GM crops has to be labeled to show this. Figure 20.11, tins of tomato puree with a label telling consumers that the tomatoes are genetically modified. Work in a small group to find out whether this is done in your country. If it is, you may be able to collect some labels with GM information on them. Then discuss in your group whether or not you think it is important that foods containing GM products should be labeled. Identify your reasons for your point of view. Your class can then take a vote on whether they think it is important to label GM foods or not. Question 12. Copy and complete this table to summarize some examples of genetic modification. Type of genetic modification. 1 example. Comments. Modifying bacteria to produce human proteins. Modifying crop plants to confer resistance to herbicides. Modifying crop plants to confer resistance to pests. Modifying crop plants to improve nutritional qualities. Advantages and disadvantages of genetically modified crops. There are some concerns about the use of genetically modified crops, and it is important that these are carefully considered. One argument against the use of GM crops is that we simply do not need them. For example, some people have argued that golden rice is not the way to solve problems caused by vitamin A deficiency. They say that it would be better to solve the real cause of these problems, which is that people do not have enough money or enough food in some parts of the world. Others argue that at least golden rice may be able to help some people now, whereas trying to get rid of poverty can be very complicated and take a long time. Another argument against their use is that the seeds are more expensive for farmers to buy than non-GM seeds. This could mean that many farmers in developing countries cannot afford to buy the seeds, which could place them at a disadvantage compared with farmers who can afford these seeds. The companies that sell GM seeds generally prevent farmers from saving their own seed and insist that they buy new seed from the company each year. A strong argument for growing GM crops that are resistant to herbicides is that it reduces the number of times that farmers need to spray herbicides onto their crops. This reduces the number of occasions on which the herbicide might cause harm to other plants growing nearby. Growing herbicide-resistant crops can also increase yields because the spray that is used is very effective at killing all plants except the crop plants. This means that there is less competition from weeds and the crop plants grow better. It also reduces labor costs, so it could mean cheaper food. Arguments against growing GM herbicide-resistant crops include a concern that the herbicide resistance gene might spread from the crop plants into other plants growing nearby, producing superweeds that can no longer be killed by herbicides. This could happen if pollen from the GM crop plant fell onto the stigma of a different plant and the male gametes inside the pollen fertilized an ovule. This is very unlikely, however, because it is very unusual for pollen of one species to be able to grow on the stigma of another species. So far, despite millions of hectares of these GM crops being grown, no superweed has yet appeared. GM crops with resistance to pests have also been a source of concern. For example, some people think that eating Bt maize may harm their health because the crops contain a toxin that kills insects. The Bt protein is a naturally occurring protein produced by a bacterium called Bacillus thuringiensis, which normally lives in soil. It is not something completely new. The Bt protein only works as a toxin when it binds with specific protein receptors that are found in the gut of the pest insects, but not in humans or other animals. Numerous tests have found absolutely no evidence that eating these crops causes any harm at all to people. It is also possible that pesticide-resistant crops might harm insects other than those that eat the crop and cause damage, for example bees that eat nectar from the flowers, or ladybirds that are predators of insect pests such as aphids. However, so far studies show little or no effects on non-pest species of insects. Nevertheless, more studies need to be done before we can be sure that this is always true. Most evidence collected so far suggests that the use of the GM crops may benefit biodiversity because the farmer does not have to spray the crop with insecticides that kill all kinds of insects, not just those that damage the crops. One issue that is emerging is that some pest insects are evolving to become resistant to the Bt toxin. This is not a new problem, as resistance to ordinary pesticides has evolved on many occasions. A possible way of avoiding this problem is to plant only certain areas with the Bt-containing crop and grow non-Bt crops nearby. Insect pests will be able to feed on the non-Bt crop, and so the selection pressure for resistance to Bt will not be as great. How genetic modification is done. To explain the processes that are involved in genetic modification, we will look at the way in which bacteria can be genetically modified to produce a human protein. These proteins are made to use in medicine to supply to people who are unable to make them in their own bodies. The first human protein made using GM bacteria was insulin, used to treat diabetes. Today, human proteins produced by GM microorganisms are widely used. Blood proteins that are essential for blood clotting are used to treat people with the inherited disease hemophilia. Human growth hormone is used to treat people who would otherwise not grow fully. Interferon is used to treat some types of cancer. Figure 20.12, how bacteria can be genetically modified to make a human protein. All of these can now be manufactured quickly and in large quantities using GM bacteria. The process begins by identifying the length of DNA, the gene, that codes for the production of the desired protein. Enzymes called restriction enzymes are used to cut the DNA on either side of the gene. These enzymes cut DNA molecules at particular points. They leave short lengths of unpaired bases at either end of the cut DNA, called sticky ends. Next, plasmids are removed from bacteria. You may remember that a plasmid is a small circle of DNA that is found in many bacteria. It is not the main bacterial chromosome. The ring of DNA in the plasmid is cut using the same restriction enzymes that were used for cutting the human DNA. The reason for using the same restriction enzyme is so that the sticky ends on the plasmid DNA are complementary to the sticky ends that were left on the human DNA. The human DNA and the cut plasmids are now mixed together. The sticky ends on the human DNA pair up with the complementary sticky ends on the plasmid DNA. Another enzyme, called DNA ligase, links the two strands firmly together, closing the plasmid loop. Now we have plasmids that contain the human insulin gene. They are called recombinant plasmids because they contain a combination of bacterial and human DNA. Next, these genetically modified plasmids are added to a culture of bacteria. Some of the bacteria take up the plasmids into their cells. These are now genetically modified bacteria because they contain human DNA as well as their own. The GM bacteria are put into fermenters where they reproduce asexually to form large populations. They express the gene for the human protein, that is, they use the code on the human DNA to make the human protein, which can be extracted from the fermenters and purified. Questions. 13. Outline the use of these enzymes in genetic modification, are restriction enzymes DNA ligase. 14. Explain the importance of using the same restriction enzyme on a plasmid and on the DNA that is to be joined to the plasmid. Summary. Bacteria are useful in biotechnology because they reproduce rapidly and can make complex molecules. Other reasons for using bacteria in biotechnology include the absence of ethical concerns and the presence of plasmids in their cells. Anaerobic respiration in yeast produces carbon dioxide that helps bread to rise. Anaerobic respiration in yeast produces ethanol, which can be used as biofuel. Pectinase is an enzyme that breaks down pectin that holds plant cells together. Using pectinase increases the quantity of juice that can be extracted from fruits, and it can also be used to clarify the juice. Biological washing powders contain enzymes as well as detergents. The enzymes usually include protease and lipase, and sometimes also amylase. They break down proteins, fats and starches that can stain clothes, making them soluble in water so they can be washed off the fabric. Many people cannot digest lactose because they do not produce lactase when they become adults. Lactase can be added to milk to break down the lactose to glucose and galactose. This makes the milk and products made from it safe for everyone to drink. A fermenter is a large container in which bacteria or yeast can be grown on a large scale in controlled conditions to produce useful products. The fermenter is designed so that nutrients and air can be added and the temperature and pH can be controlled. Waste products such as carbon dioxide are removed. Genetic modification involves changing the genetic material of an organism by removing, changing, or inserting individual genes. Bacteria have been genetically modified by inserting human genes for making proteins such as insulin and human growth hormone, so that these proteins can be produced on a large scale for use in medicine. Crop plants have been genetically modified to make them resistant to herbicides, so that spraying a herbicide on the crop kills weeds but not the crop plants. Crop plants have been genetically modified to make them resistant to insect pests, so that the farmer does not need to spray pesticides on the crop. Rice has been genetically modified to produce more carotene, which helps to prevent blindness or death resulting from vitamin A deficiency in people whose diet contains mostly rice. There are arguments for and against the use of genetically modifying crops such as soya, maize and rice. The first step in genetic modification of a bacterium to make a human protein involves cutting out the required human gene with restriction enzymes, leaving sticky ends. Bacterial plasmids are cut with the same restriction enzymes, leaving complementary sticky ends. The gene and cut plasmids are mixed so that the complementary sticky ends join together. The strands of DNA are joined using DNA ligase. This forms a recombinant plasmid containing both bacterial DNA and human DNA. The recombinant plasmids are inserted into bacteria. The bacteria multiply to make a large population of genetically modified bacteria. The genetically modified bacteria express the human gene and synthesize the human protein. Project: Debating the use of genetically modified crops. You are going to take part in a debate about whether your class agrees or disagrees with this statement. This class believes that growing genetically modified crops does more good than harm. Half of your class should help to construct arguments to support the statement. The other half should help to construct arguments against the statement. You may be able to choose which side you are on, but be prepared to have to argue for a point of view that you do not agree with. The aim of the debate is to help you to organize your ideas and to understand the arguments for and against the statement, so even if you have to argue for something you do not believe in, you will gain from the experience. Figure 20.13 Students taking part in a debate. Structuring the debate. The first speaker for one of the sides presents their team's case. They should put forward 2 or 3 strong arguments, with supporting evidence if necessary. The first speaker for the other side now does the same. They should use their prepared arguments but should also try to rebut whatever the first speaker said. Next, the second speaker for the first side presents their case. Next, the second speaker for the second side does the same. Things for your class to decide on before you begin. Depending on the size of your class and how long you have, you could also have a third speaker for each side. You should limit the length of time that each speaker is allowed. This will depend on how long you have, but in general about 4 or 5 minutes will be sufficient. Will you allow people to ask questions of the person who is speaking? Structuring your arguments. Here are some things to think about as you build your presentation of your case. Start with a clear statement about your argument, which says exactly why you are for or against the statement. Next, present some evidence for your point of view. Finish by explaining how the evidence supports your claim. Make sure that the second speaker for each side uses arguments that are not just repeating those of the first speaker. Here are some things to think about as you think of ways of rebutting an earlier speaker's arguments. How strong is their evidence? Is it just a few isolated stories, or is there good scientific research behind it? Have they confused correlation with causation? Just because 2 things seem to be linked, it does not mean that one causes the other. Are things really as simple as the speaker suggested, or is it more complicated in real life? The final vote. When each speaker has finished, the class should vote on the original statement. At this point, you are free to vote however you wish. You can vote against the argument that your team was putting forward, if you want to. Exam-style questions. 1. What is pectinase used for? A. Digesting food for babies. B. Producing carbon dioxide for bread, making obtaining more juice from fruit. D. Removing stains from clothing. 2. What is a reason for using bacteria in biotechnology? A. They can cause disease. B. They do not photosynthesize. C. They have cell walls. D. They reproduce rapidly. 3. What is an example of genetic modification? A. Crossing two animals with different genotypes together. B. Putting a gene from one species into an organism of a different species, selectively breeding cattle to produce more milk. D. Using yeast to make biofuel. 4. What is used to join two DNA strands together during genetic modification? Amylase, ligase, pectinase. Restriction enzyme. 6. Yeast is used in bread making. A. I name the process that occurs in yeast cells which produces a gas that helps the bread to rise. 2. Name the gas that is produced by yeast during bread making. 3. Name one other substance that is produced by the yeast during this process. B. Yeast is also used for making biofuel. I. What is biofuel? 2. Describe how yeast is used to make biofuel. 7. A. Outline one example of using genetically modified bacteria to produce a human protein. B. Suggest one advantage of using bacteria to make this protein. C. State two differences between selective breeding and genetic modification. 5. What is not an advantage of inserting genes into soya plants to make them resistant to insect pests? A. Higher yields are obtained. B. Less insecticide is used. C. Nutritional quality of the crop is improved. D. Populations of insect pests are reduced. B. Undigested lactose is usually broken down by bacteria in the small intestine. This produces hydrogen. Testing for hydrogen in the breath after consuming lactose is one method of diagnosing intolerance to lactose. Explain how hydrogen produced in the gut can become a component of exhaled air. C. Lactase can be made commercially using the bacterium Lactobacillus. The graphs below show how temperature and pH affect the quantity of lactase that the bacterium synthesizes during the time the bacterium is grown in a fermenter. 8. Lactase is an enzyme produced in babies which digests lactose in milk. The enzyme is present in the cell membranes of the cells covering the villi in the small intestine. In most adults, lactase is not secreted. People who do not secrete lactase may suffer from gut pain and diarrhea. Undigested lactose causes an increase in the water content of the gut lumen. Suggest how this is caused. Use the information in the graphs and your own knowledge to suggest how lactobacillus should be grown in a fermenter and explain how optimum conditions for its growth should be maintained. 9. This graph shows how the use of two genetically modified crops changed in the USA between 1996 and 2019. A. Compare the changes in the use of herbicide-resistant soya and pest-resistant maize. B. Explain why growing maize that is resistant to insect pests can be advantageous to both the farmer and the environment. C. Evaluate the advantages and disadvantages of growing herbicide-resistant soya rather than non-GM soya. Self-evaluation checklist. After studying this chapter, think about how confident you are with the different topics. This will help you to see any gaps in your knowledge and help you to learn more effectively. I can. See topic. Needs more work. Almost there. Confident to move on. List reasons why bacteria are useful in biotechnology and genetic modification. 20.1. Describe the role of anaerobic respiration in yeast during production of ethanol for making biofuel. 20.1. Describe the role or anaerobic respiration in yeast in making carbon dioxide to help bread to rise. 20.1. Describe how pectinase is used in the production of fruit juice. 20.1. Investigate and describe the use of biological washing powders. 20.1. Describe genetic modification as changing the genetic material of an organism by removing, changing or inserting individual genes. 20.2. Outline examples of genetic modification, including using bacteria to make human proteins, making crops resistant to insect pests or to herbicides, improving the nutritional qualities of crops. 20.2. Discuss why bacteria are useful in biotechnology and genetic modification. 20.1. Explain how and why lactase is used to make lactose-free milk. 20.1. Describe and explain how bacteria and yeast can be grown in controlled conditions in a fermenter. 20.1. Describe how genetic modification is carried out with reference to sticky ends, plasmids, restriction enzymes, DNA ligase and recombinant plasmids. 20.2. Discuss the advantages and disadvantages of genetic modification of crops, including soya, maize and rice. 20.2.